you so much for being here. Wow. Okay. Hello, everyone. My name is Gihoon, and I'm the CEO and co-founder of SHIFTBIO, a biotech company based in Seoul, South Korea. Our company aims to bring the next paradigm shift in new drug development using AI-driven natural drug delivery platform technologies. Before we start, I want to introduce our team member first, and then I will show you how virtual twin technologies advance the drug delivery systems. Our company was founded by 3 MD PhD co-founders, so we have a deep understanding of patient urgency and unmet medical needs. And our key co-founder, Professor In-Sang Kim, is a renowned figure in the field of nanomedicines and drug delivery system. He is one of 2 scientists honored with government protection due to his invaluable scientific capabilities in South Korea. He has the crucial role and principal technologies at this company. And our company has 2 CEOs, and our CEOs has the special nicknames. The one sleeping in South Korea right now is the Chief Empowering Officer. Look at this muscle, so empowering and powerful. So if you're interested in him, please let me know. He is single. The other one, which is me, I'm the Chief Everything Officer. I play the MacGyver at this company. So the best part about startup is I can learn everything, but the worst part about startup, I have to handle everything. So anything practical regarding SHIFTBIO, I'm your guy. Feel free to contact me anytime. But as you know, company success isn't powered only by leaderships. Especially, even we have the special CEOs. Our company is driven by an amazing team of 23 members. More than half of them are scientists, and we have also guided by an exceptional scientific advisory board, including Professor Tom Roberts, the former dean of Harvard Medical School. We believe that we have strong scientific capabilities, and we also believe that strong science can make breakthrough and can bring the next paradigm shift in new drug development. With these strong scientific capabilities, we have dedicated to focus on only one thing, that is drug delivery. Yes, we admit it, we are crazy for drug delivery. So why are so obsessed with drug delivery? Because behind many unmet medical needs lies what we call the delivery void. In other words, we have promising cargoes, but we don't have right vehicle to deliver it. Although numerous therapeutic cargoes and targets have been discovered for decades, most of them have failed due to the limited delivery efficiency. First problem is biological barrier. The blood-brain barrier blocks almost everything, 98% of small molecules and almost 100% of biologics like proteins. That is why numerous promising drugs cannot reach patients suffering from brain diseases. The second problem is fragile cargoes, that means fragile drugs or modalities. Take siRNA, very promising genetic materials and genetic medicines, for example, but on its own, it's useless. When drug delivery tool came along, siRNA became a drug. This is how the first siRNA drug was born. So we want to deliver better than anyone else. To achieve this, we go beyond the synthetic drug delivery system like lipid nanoparticles or virus-like particles or antibodies. So we want to fully leverage the cell-to-cell communication system in our body, cell-derived nanoparticle, or called extracellular vesicles. And we can create these extracellular vesicles more qualified, and we are calling this the natural nanoparticle NMPs. These NMPs can offer low immunogenicity and great targeted delivery efficiency with scalable manufacturing and high loading drug capacity. Today, I will explain and talk about how we redefining these natural nanoparticles with digital twin technologies. But before we move on, let me ask you one simple question. Do you know how much it really costs to develop a single drug? Have you know that? Do you know that? Yeah. The answer is $2.3 billion. And it takes over almost 10 years to bring one successful drug to patients. But the problem is that is on average. So for advanced medicines like cell and gene therapies, the cost is much, much higher. Yes, I'm the founder of a new drug development startup for advanced medicines. With the level of this risk, am I a gambler? Maybe I am. That is me in a casino at Boston, nearby here. Perhaps I am a little too sensitive to dopamine. However, the point is that, here is the point, the success rate of new drug development is far lower than a winning a gamble as a professional player. So to win this reckless gamble as a small biotech companies, where a single mistake at any stage means not a delay, but extinction. So I have to do more than play better. I should change the rules themselves. As you know, and everyone knows that drug development can change patient life and also create a substantial value. But this journey is also high-risk game. So how can a small biotech companies like ours can take on such challenges? We found this answer in digital twin technologies. And we believe that these virtual twin technologies can make lower risk business model for new drug development. And this is where we stop betting on luck and start engineering certainty. 3 years ago, we met Dassault Systèmes and Bernard Charlet. And yeah, this meeting has changed everything. We had an inspiring conversation. And since then, we have started to imagine the incorporation of virtual twin technologies into our natural drug delivery platform technologies. And we also met fantastic BioVia research team, where I asked Dr. Deepa, can you make my dream come true? I remember her smiling and saying, yes, I can. This is another photo with Pascal CEO, who reaffirmed our company's journey is possible together with Dassault Systèmes. So our journey would never have been possible without the technologies of Dassault Systèmes and unwavering their support. With these partnerships and their collaborations, we are now moving forward to turn our dream into reality. So first, I'd like to take this moment to express my deepest gratitude to Dassault Systèmes team. From now on, let's move on to the main part of today's section, the AI-driven natural drug delivery platform technologies. The current drug delivery system faces significant limitation in targeted delivery, immunogenicity, and drug loading capacity. Extracellular vesicles or cell-derived nanoparticles have emerged as a promising solution to address these challenges. However, these extracellular vesicle therapeutics also have major bottlenecks in design complexity and resource-intensive manufacturing. That means very high-risk business drug development. So to tackle these issues, we have created an innovative approaches to transform traditional extracellular vesicles into clinical accessible, the next generation drug delivery tools. We are calling this qualified extracellular vesicle as a natural nanoparticle NMPs. This is just our brand name. There are two core approaches. The first one is NMP Design AI platform to de-risk and streamline designing this nanoparticle using in silico modeling system before well-lab experiments. Second, NMP manufacturing approach to produce the high purity and high quality and very efficient cost natural nanoparticle using GMP-compatible manufacturing process, scalable manufacturing process. These nanoparticles are biologics and released from cells. So our approaches reprogram cells to generate the specific drug delivery tools with targeting moieties. Our team has created our own engineering scaffold. We are calling this the Natural Nanoparticle Sorting Motif, NSM. This NSM is our own amino acid sequences-based scaffold and very short transmembrane domain motif, which can be effectively incorporated into the bilayer of cell-derived nanovesicles in the cells during their biogenesis. By leveraging this NSM scaffold, we can decorate the surface to express the targeting moieties, and we can load the macromolecules into the inner cavity of these nanoparticles. And this NSM has three distinctive advantages. The first, high loading capacity. Second, multiplex cargo loading. This means that we can load several cargos into single nanoparticle. Third, universal cell application. We already verified that these techniques can be applied to diverse cell lines or primary cells such as stem cell and immune cells. This is one example to verify our competitiveness. We already confirmed that our NSM scaffold outperformed the current competitive scaffold in the aspect of loading therapeutic cargos and expressing the targeting moieties on the surface. But what truly sets us apart from competitor is that we understand the mechanism of how NSM can go into the nanovesicles during their biogenesis. This is the key point because based on this mechanism, we can incorporate the in silico modeling system into our platform technologies. So we understand how NSM can get into the nanovesicle during their biogenesis in the cells. We can explain this mechanism through the protein-protein interactions. Specifically, we have identified the specific proteins such as tetraspanines, CD9, CD81, CD16, that play a crucial role in this entire engineering the ones that effectively interact with this NSM scaffold. So our next question is, how can you streamline this engineering process? Because this is biologics, so we want to control and de-risk this engineering process too. So our answer is virtual twin. If we can predict the structure of NSM, and if we can analyze this protein-protein interaction between NSM and tetraspanin, which is core for this engineering, we hypothesize that we can pre-access this engineering efficiency and designing efficiency before experiments. So we have collaborated with BioBia research team for 3 years, and specifically using the 3D experience platform, we first predict the multimer structure of our NSM cargo with the core protein tetraspanins. After refining structures with minimization and implicit membrane, and then we perform the protein interaction analysis, and we identified the key binding site in these interactions. And then we perform the molecular dynamics to identify the key residues on this interaction. So based on finding these key residues and key binding site of NSM and these key proteins, we can calculate interaction energy on this binding site. And surprisingly, we finally demonstrate that this scoring, this interaction energy is closely correlated with the real engineering efficiency data in the cells. So this means that if we can calculate the interaction energies, NSM and these specific proteins in silico modeling system, we can predict the engineering efficiency of the cells to generate this nanoparticle before experiments. But I want to emphasize one more thing. The point is that we realized that this virtual data is not just for simulation. It's also real data. For example, this is just an example. So in the left part, looks like very simple, but this is the general biological data. But it requires 1 year to complete this data set. Because, we have to create the stable cell lines and establish SOP and check the reproducibility and generate the repetitive data to analyze the statistics for this data. So this data set, we need 1 year to complete these things. But in the right part, we just used the 20 minutes to generate the same amount of the data in virtual simulations, just 20 minutes. So we know the mechanism, how NSM can move into those cells and move into the extracellular vesicles. So we already confirmed that this expression level and biological data can closely correlate with in silico data. So this virtual data can be used for real data, and we can make a lot of data to drive the new ideas and creative hypotheses to advance our drug delivery systems. So through this collaboration, we finally built NLP Design AI to de-risk and to streamline this engineering process. And we believe that this can significantly reduce cost and time for making ideal drug delivery tools. So next part is our manufacturing approach. We recently published our manufacturing achievements in Nature Communication papers. And we successfully scaled up the production of large quantities of natural nanoparticles with high yield. And we have optimized the downstream process to achieve over 95% purity and over 50% recovery rates. These figures are really impactful in this field. And we are confident that our manufacturing approach is highly transferable, and we are ready for clinical manufacturing of these nanoparticles with reasonable cost. However, even with our strong manufacturing capabilities, the customizing production for each drug delivery tool remains a major cost bottleneck. From our experience with Dassault Systèmes team in drug design, we realized that the same approach on virtual simulation can be also applied to this manufacturing approach. We now envision expanding our capabilities further by incorporating the additional solutions, such as Delmia, into our manufacturing approach to advance effective manufacturing cost or process. So leveraging these NMP design and manufacturing approaches, we have showcased the two targeted delivery solutions. The one is the DTBB NMP for systemic delivery of payload to the specific brain site through the intravenous injection. And second one is SERP NMP for targeted delivery to inflammation site for the treatment of autoimmune diseases and fibrotic conditions or inflammatory diseases. To achieve the brain targeted delivery, we highly expressed our own dual targeting brain shuttle on the surface of these nanoparticles. As designed, the intravenous injected this nanoparticle can really cross the blood-brain barrier and can reach out the brain parenchymal cells such as neuron. And surprisingly, this can enable deliver macromolecules like proteins or oligonucleotides to specific brain cells through the systemic delivery. Regarding our inflammation-targeted delivery, SERP NMP is designed to effectively accumulate at the site of inflammation. So this can target tumor tissues and fibrotic tissues and autoimmune disease pathological lesions because these have the same common pathological mechanism, that is the inflammation, and we found the universal target of these diseases, so our SERP NMP is really effectively targeted to these tissues through the systemic delivery. Based on this drug delivery tool, we have developed multiple therapeutic pipelines to address high unmet medical needs in oncology, neurology, and inflammations. And particular, our first program, SBI-102, is on track to enter a clinical trial next year. This is another de-risking new drug development strategies. We are now engaging over 15 multinational companies, 50+ multinational companies for research collaboration to develop new candidates using our drug delivery platform technologies. And we are confident that we are on the verge of making meaningful partnerships with global partners. But despite in contact with many companies, we are still physically limited because we are small biotech, so only carry out 1 or 2 projects at a time. We believe that virtual twin can expand our capabilities to make meaningful deals with global partners, and this means that this can multiply the value of our platform technologies. So from the drug design and manufacturing and virtual twin simulations can enhance our capabilities to develop the new drug development. And this means that we can make several pipelines to give a new chance to patients with limited resources. I believe that AI is no longer optional. AI is essential in new drug development. We completed NMP design AI with digital twin technologies. This is a very small start, but we want to expand this approach on the whole drug development of our drug delivery systems. We believe that digital twin technologies can de-risk and streamline each step from target identification and NMP design and NMP manufacturing and clinical trial. Right now, we established the manufacturing approach, and also we established the target identification tools. So we are start to discuss the incorporation of digital twin simulation into our approaches to make promising drugs and to establish the whole drug development ecosystems. So this digital twin approach is what allows us to make the innovation of NMPs into reality. And we also think that this approach is not just a process, it's a product of transformative framework for future drug development. But still some say, why natural nanoparticles? And still they say, new modalities won't work in today's biomarket. But for me, those perspective is too boring and don't leave much of an impact anymore. We know that history echoes itself. In the 1990s, people didn't believe antibodies' drug abilities. In the 2000s, antibody-drug conjugate were considered as a bundle of toxicity. And in 2010, cell therapy were thought to be impossible due to their heterogeneity. In 2020, mRNA leading company went to almost bankruptcy before COVID-19. But we know that these modalities turn out to be game changer in new drug development market. History repeats itself, and the time for new innovation, I believe that it's just around the corner. The unchanged truth is that this natural nanoparticle is a promising modalities and innovative drug delivery system. So we must ask ourselves again is, what we can solve the problem using these promising modalities? And how can we change the patient's life using these new techniques? We choose natural nanoparticles to create the new medicines, not because they are easy, but because they are required. Because that core will change substantial impact on new drug development, because that challenges will give a new chance to patients who have no therapeutic options. We believe that with digital twin technologies, we are confident that we can make innovation accessible from science to patients. For the loved one, for the family, for the patients, we want to bring the next paradigm shift in new drug development using our AI-driven natural drug delivery platform technologies. Thank you for listening to my presentation. Thank you.
Wow. Han, I get to understand two things. Now I know why you are the empowerment chief officer. Now this is very clear. And I understand why SHIFTBIO is the next paradigm.
Thank you. Yeah.
So let's open for question in the room. Any question for Han so far? If not, I will start. It's very, very compelling, the vision that you have and how you are, of course, leveraging our solutions all along the life cycle. Are you intending to change the design with the manufacturing return of experience?
Oh, yeah, very good question. Yeah, I think that's possible, and because I'm not sure about it exactly, but we are feeling that the virtual simulation and virtual twin is not just simulation. It's also real data, or I'm not sure what is the appropriate word right now, but so I think that a combination of virtual and real data, and based on the strong mechanism, and we can make some new things in manufacturing design, yeah, effectively.
And in your pipeline, you mentioned agent at the very beginning. Can you expand a little bit on that?
So yeah, that is a little another story because we are using public single-cell RNA sequencing data, and we are really focusing on the spatial transcriptomics data because, as you know, when we use the bulk RNA sequence data, that is just average level of specific genes, but move on to the single-cell RNA sequence data, we can find out new targets because we can add one dimension.
That's the agent.
Yeah, and spatial transcription, one more dimension, but very big data. But we found that, so as you know, one disease has many stages, and at each stages have the distinct cell-cell interaction.
Yes.
And we want to find out a new master regulator of target based on this cell-cell interaction because we can deliver macromolecules and, genetic materials effectively. We can incorporate target identification and drug design using this platform.
Understand.
Yeah.
Understand.
But the reason we're using AI agent because we want to make user-friendly environment to our scientists like me. So yeah, so that is my point. Yeah.
And you mentioned NNP AI direct design and NNP manufacturing, but no AI. Are you also envisioning to add the AI dimension in the manufacturing part?
Absolutely, but this is just our future things and to-be things, but yeah, easily we can simulate the process using Delmia, but I think that there is some more and more things like we envision that we can also generate the manufacturing data.
Absolutely.
Yeah, to demonstrate the batch-to-batch consistency because
Batch. With the batch.
this requires a lot of costs, especially in small biotech. So yeah, we already start to think about this thing, and we collaborate with these things with Korean Dassault Systèmes team, and we understand the 3D experience platform for manufacturing, and we have some hypothesis to incorporate these techniques into our platforms. Right?
This is very impressive. I have the feeling that not only SHIFTBIO is the next paradigm for treatment, but also for using Twin as a next lever and catalyst.
Yeah.
Let's really, there's a question over here.
One question. Thank you so much for a great talk. NNPs clearly are going to change the game. What class of targets do you consider to be ideal for NNPs, which today we may have the cargo for, but not the ability to deliver the cargo with?
Yeah, very good question. So I can respond this question in two ways. The first thing is about the targeting side. This is a little business reason, but this is also high on my medical needs. We are really focusing on brain tissues because, yeah, there are some antibodies can cross the BBB, but as you know, normal antibodies can enter the brain tissue through the intravenous injection just 0.1%. But now techniques can enhance this delivery efficiency around 1%. But we already have data that our natural nanoparticle can outperform this brain-targeted delivery efficiency, and many multinational companies want to have the new delivery tools to address the brain diseases. So we are focusing on brain science right now and autoimmune diseases too. And the second way is the cargos. So we are focusing on macromolecules like proteins and genetic material because macromolecules is really unstable in our body system. So we believe that these cargos require optimal drug delivery system. And as you know, genetic material is also unstable in our bloodstream. So we believe that the low immunogenicity drug delivery tool, natural nanoparticle, can have strong advantages to deliver these cargos effectively. Yeah.
Thank you very much. If there are no other questions, let's really give a warm round of applause. Congratulations, Anne, for your remarkable presentation. Thank you so much. Thank you so much.